Namaskaram. My name is S. Ashok Kumar. Welcome to my channel. Now, in the last part, I explained about the monosaccharides. Now, in this part, I am going to explain disaccharides. Disaccharides are the most common oligosaccharides. Okay? That is why explaining the disaccharides. Actually, disaccharides are classified into two types. One is a reducing disaccharides. Next one is a non-reducing disaccharides. Actually, there is a different ways of telling reducing and non-reducing. Here I am telling in an understandable way. Reducing means a reactive. The reactivity of the molecule due to the presence of free functional group. That I will explain. The reducing disaccharides, examples, maltose and lactose. Here comes to the non-reducing. Non-reducing means what? Non-reactivity. That is because of the absence of the free functional group. Example for non-reducing disaccharide, that is a sucrose. Okay? So here what I said, reducing means reactive. So disaccharides divided into reducing and non-reducing. What about monosaccharides? All monosaccharides, in all monosaccharides, functional group is a free. That means they can react. Can react we are calling as a reducing. That is why all monosaccharides comes under reducing sugars. Maltose and lactose belongs to reducing disaccharides. Now first I am going to start with the reducing disaccharides. Reducing disaccharide. What are the examples for reducing disaccharides? Maltose and lactose. First I am going to start with the maltose. Maltose, malt sugar, maltose. What is this actually chemistry that I am going to explain the structure of the maltose. Maltose consists of two molecules of alpha D glucopyranose. Up to here you remember. I will explain the remaining thing. Maltose consists of two molecules of alpha D glucopyranose. Bond name that I will give the clarity. So first alpha D glucopyranose. See, very simple way. Pyranose, alpha D glucopyranose na 1, 2, 3, 4, 6 CH2OH. Okay. Alpha na OH group is at down. Remaining I am showing here. This is a alpha D glucopyranose. Maltose consists of two molecules of alpha D glucopyranose na. One more molecule I have to write. Same thing I am going to write here. Alpha C CH2OH. Okay na? OH, OH, so 1, 4. This is also alpha D glucopyranose. You have to concentrate on the board. In the disaccharide formation, what is happening here? Maltose formation, first carbon, that is nothing but functional carbon of this sugar. Here having the OH group. This is the second sugar, this is the functional carbon, but this is the fourth carbon. Here OH group, here H group. Removing as water. Removing of water is called as a dehydration. Dehydration. Now understand the maltose structure. See, same thing will come. What is the difference? You need to understand. This is the first carbon. Here remaining oxygen. Okay, first carbon. Now, this is the fourth carbon. This is the fourth carbon. Remaining I am filling here like this. CH2OH. See, this line indicates the OH group. This structure is called maltose. This structure is a maltose. Now I will explain. Here This, before going to explain this one, what is the name of this bond? Okay. Here, this one, any bond that links with a sugar, what we know, that is a glycosidic bond. What is the name of the glycosidic bond present in the maltose means the first sugar is belongs to alpha sugar, na? That is a alpha. From which carbon to which carbon bond is formed? 1, 2, 4 glycosidic bond. Okay. So how can you define maltose completely? Maltose consists of two molecules of alpha D-glucopyranose residues linked through alpha 1 to 4 glycosidic bond. Here, the next point I am going to explain, when the glycosidic bond is forming, there is a release of water. Na? Releasing of water is called as a dehydration. That is why second point I am telling, glycosidic bond formation is example of dehydration. The same thing I repeated in so many times. Peptide bond formation is an example of dehydration. Phosphodiester bond formation is an example of dehydration. Glycosidic bond formation is also an example of dehydration. Okay. So, this is the disaccharide is formed as a result of dehydration. That is why the product formed as a result of dehydration, that product we have to call it as a residue. That is why what is the generalized definition of disaccharide? Disaccharide consists of two monosaccharide residues linked by one glycosidic bond. That is a generalized definition of disaccharides. Here, maltose belongs to reduced sugar. Na? What I said the reducing sugar means? There should be presence of free functional group. Yes or no? Now understand. There is here the first sugar, this is a functional carbon. The functional carbon on the first sugar is engaged with the glycosidic bond. So there is no free functional group on the first sugar. But when it comes to here, this is a functional carbon on the second sugar. So this functional carbon is a free. Na? That is why the reducing nature of maltose due to the presence of free functional group on the second sugar. This is a way of understanding the disaccharide. I explained by taking the maltose. Remaining disaccharide I will explain soon. After explaining the structure of maltose, now I am going to explain lactose. That is also belongs to reducing disaccharide. This lactose commonly called milk sugar. Why? Because it is present in the milk. So lactose is a milk sugar. What is a milk protein? That is a casein. The next one, the enzyme causes breakdown of lactose is called lactase. This is everybody tell. This lactase is also called as a beta-galactosidase. Beta-galactosidase enzyme produced by lag-EZ gene of lactoperon. This point important. The enzyme causes the breakdown of lactose is called lactase or beta-galactosidase. This is there in the molecular value, simultaneous in the biotechnology also. There is a concept called as insertional inactivation. There you will get the enzyme about beta-galactosidase. Now go to the structure of the lactose. This lactose consists of beta-D-galactopyranose links with beta-D-glucopyranose. I will make you understand. First you take the beta-D-galactopyranose. This is a pyranose, 1, 4th carbon. Beta na OH group is a upside. Galactose, right? Carefully understand. CH2OH. This is the 6th carbon. OH, 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 OH. 4th carbon OH group also upside. This is a beta-D-galactopyranose. Now links with the beta-D-glucopyranose. Say similar structure. 1, 4. Beta OH group is a upside. Glucose na C. Here you can clearly discriminate between the glucose and galactose. Okay na? OH is this. This one is a beta D glucopyranose. Simple one. In the first sugar, that is a galactose, first carbon OH group upside, functional carbon. In second sugar, that is a glucose, fourth carbon OH down. So here OH, here H. Removing as a water. What I said, glycosidic bond formation is an example of dehydration. The resulting structure, what is the change you need to understand? See, up here oxygen remaining, C. Beta, this is a glucose, CH2OH. This is a galactose, CH2OH. This is the first carbon, this is the fourth carbon. Fourth carbon, first carbon. Yes or no? What is the name of this bond? First sugar which anomer? Beta na? So beta. From which carbon to which carbon? 1 to 4. Glycosidic bond. So what is the bond in maltose? Alpha 1 to 4 glycosidic bond. What is the bond in the lactose? Beta 1 to 4 glycosidic bond. One more conclusion what you need to understand. This lactose belongs to reducing sugar na? The reducing nature due to the presence of free functional group. And the second sugar that belongs to glucose. That is why maltose and lactose both are belongs to reducing disaccharides. After reducing disaccharides, now I am going to explain non-reducing disaccharide. That example is a well-known sugar, that is the sucrose. This sucrose is called as a table sugar. Table sugar, the one we are using, also called as a cane sugar. This is the one point. Next point, the enzyme causes the breakdown of sucrose. That is called as a sucrase or invertase. The enzyme causes the breakdown of sucrose is called as a sucrase or invertase, because the inversion is one of the very important property of the sucrose. Comes to the structure. What is the sucrose consists of? Sucrose consists of alpha D glucopyranose links with beta D fructofuranose. Here in the sucrose, the very important thing is the bond. Now first you take the alpha D glucopyranose. This is a structure you should practice because glucose will come so many times. 1, 4. Alpha na OH group down. CH2OH. OH, OH, OH. This is a alpha D glucopyranose. Okay na? Next sugar, beta D fructofuranose. To get it very quickly to understand to you, that beta D fructofuranose I am writing here. See this one. Same thing I wrote in the monosaccharides itself. 2, 3, 4, 5. Okay na? Beta. OH group is the upside. First carbon is a CH2OH. First carbon. Then OH. OH. Sixth carbon CH2OH. This one is the beta D fructofuranose. Okay. Now we are going to make the bond. Here what is happening? The very important thing, in glucose functional carbon first. In fructose functional carbon is the second. So in glucose functional carbon OH group, in fructose also functional carbon OH group. So here OH group, here OH group. Removing as a water. Okay, na dehydration. Now the resulting structure, simple. First carbon. Oxygen, remaining oxygen is there na? Next one, see. This one is a down, one is a CH2OH. CH2OH. This structure is a sucrose. Now, very important thing is the bond. What is the name of the bond present in the sucrose? See this one. This is the first sugar which is anomer alpha. Then from which carbon? Number 1. Bonds with which carbon? This is a second carbon. alpha 1 to 2 glycosidic bond. So what is a very important thing regarding the bond in the sucrose, in the sucrose, this first sugar functional carbon, in fructose second sugar functional carbon, both functional carbons simultaneously involved in the glycosidic bond formation. Then what you need to understand, if you take the overall sucrose, is there any free functional group? Absolutely no, because there is no free functional group. Where those free functional groups went? These both functional groups are simultaneously involved in the glycosidic bond formation. That is why in the sucrose structure there is no free functional group. When I said there is no free functional group, what you need to understand? Because no free functional group, this is belongs to non-reducing. Non-reducing means what? Non-reactive sugar. So how sucrose become a non-reducing sugar? Because there is no free functional group. Now the very important aspect, why I said the structure of the sucrose, because sucrose we came to know it belongs to non-reducing or non-reactive sugar. What is the importance of such a non-reactive sugars, disaccharides in the biological system? That is a very simple thing, is like in plants, actually the storing form is a starch. But starch cannot move from one place to the another place in the plant. Why? Because starch is very complex structure. That is why when the sugar want to transport in the plant, that starch is breakdown in the form of sucrose. This is sucrose, because it's a non-reactive nature, non-reducing nature, can transport from any part of the plant to the other part. That is why what is importance of the sucrose? Sucrose is the most common transportable sugar in plant, that is evolution selected because of its non-reducing nature. So till now I explained about the disaccharides. If you like this concept, you can share these videos to your friends. If you have any doubts about the chapter biomolecules, you can comment below. Okay, definitely I will respond to your questions. Subscribe our channel. Thank you.